like wear normal clothes tonight. You know what I mean? I'm excited with my dreams. I'll do tight clothes.
Yeah, I was gonna look at pink when I was out here or something. I think there's something like everything. And then I literally still grabbed black thing. Like, you know what? I don't care.
I know, I was just excited to wear my glasses and have my hair. You know?
Yeah. Just feel like a normal human again.
Yeah. And not just like,
I really go, so
I love being in art. I love it.
Oh, I had like 3 off days.
Yeah, that's true.
So like, well, like one is Sunday.
But like today's not any different.
Yeah, like yesterday I like could not go to practice at all. That sucks too, because now like I feel like this is so much like, I really can't do anything about it.
Yeah.
So I was in my freaking 3 labs.
Well, because I can look at it after going to go last night. And we saw you guys all went.
And we literally only went because, well, I wasn't even supposed to go. And I was sad. And then I just got my bus. I was like, okay, whatever. And so I was so tired. I was so tired. And we showered and I met.
It was like about 7:30.
How?
I'm so tired.
My body would not even be able to.
That's what Abby says.
Like 7:30 p.m. Like,
Yep.
what was I doing until there? I was eating dinner.
Jen was making dinner, nothing. And I was eating.
She's probably like,
Oh, we get so mad. She's like, I want to hang out.
I just probably like, where's my roommate? That's what I'm coming back.
But that's why we decided not to go.
Yeah.
Too late for us. Even for her, she's like, that's too late.
Well, that was not very strong, but it was like some kid on the football team's like birthday yesterday or 2 days ago. We just put it in and it was just all like, oh, and then they were like, we're going to celebrate him. It was that time you know, I felt really bad. And I was like, it's gonna make me like feel so bad.
Like,
So I
like sadness.
I just talked to my professor for like the last 40 minutes.
Oh, but
I went in there to get like extra help because we have a quiz tomorrow.
your genetics professor?
No, Ordo. Yeah. We just took a personal back. Genetics? Yeah. Yeah. Yeah, all season, mind you. Yes, it was so fun. No, but I could not
ever do it in college, though. Like, if I passed college.
That's what we were talking about.
If I passed
Yeah, how is he coming to me? Because that's a hard part to have a hard and he's like, oh, you'll switch it. No, I always emphasize to be good. Like, no, apparently not. Apparently, one of the evaluations we were going to do after mine, and like, we used to learn that initiative, you know, and parents, and so then it was like, I believe ecology, and then he got it and was like, I don't teach ecology, but there was one that was like,
Yeah.
did not say anything about ecology, he's like,
So
I
what
think
in
it's
groundwater?
professor, like I did, like I already taught.
The first door.
It's about you, and I was like, it's definitely not about you. He talks about his children all the time. Well, it apparently goes to like someone in like registrar building, and then it's like and they're all
Such a good thing to use.
and then it's like
Oh, I have a picture of it. No, he's actually like really did this semester.
really? I did not want to do that at midnight last night, so I did. I like light cats, but I dislike them.
Yeah, this is the pictures I can bring.
I don't know.
Like last time we get to practice.
Oh my god,
This is institution like a form like
I just don't know if anybody can stop.
But there was this one girl, my honest friend, she was like, yeah, I was busy. No, but one girl had sent her like bio to lab, and she was I know in bio 2 I did not talk about trees. So he's just in the ecology class and they're like, yeah, because he like and I'm like, that would make sense. That's like kill me. Oh my god. That was so bad. I was like, oh my god. I was leaving one person. I agree, and I believe that. So any difference, I think it's really yeah, Laura, I don't know if it was like previous years. She was like balding every week. I mean, who wouldn't? That would be crazy. I mean, okay, wait, rip damn it. Pause, go back. Go back, all the way back. Those are
What time? Yeah.
Brian, you want to read Brian's quote?
No.
No.
She's asking.
I do. I want to.
I love that open I just don't think I said that. What? After that, I just don't think it's I was trying to like, after I scared. After today, how do you not see it that he's a man? That's how he doesn't see it. I tried snapping after I was like, good vibe. And then I snapped. I was like saying, I was like, I completely misread that.
Well, good afternoon, everyone. Hey, the computer's actually working today. Yay. Hey, the recording system's actually working today too. Yay. Life is good. All right. Speaking of good things to come, have you looked ahead in the schedule to see what's coming up on Monday? Yay! Santa coming up on Monday! We're actually doing really well. We're actually like on schedule. We're on track. So we'll be able to. We'll be very close to finishing chapter 4. Today, if we will finish by Friday for sure. I'm hoping to actually put together a practice test and do a little bit of review on Friday. So that's the plan for this week. And then we'll have our in-person paper test on Monday, like in class. So we're looking at, as far as the format goes, we're looking at things like a matching. For example, like if you see something like this, you may be asked to kind of match the name of the category like the psychrophiles with the appropriate temperature in which they grow. Like that'd be kind of like a matching kind of question. You may have some fill in. Do you like fill in the blank? I am pretty generous with spelling, so don't worry about spelling. But we might have some fill in the blank. So kind of review your terms. And let me think, what else can we do here? Multiple choice. Multiple choice is always a good one.
Are you gonna post a study guide at all?
No. So the idea is that use your notes. Use your notes as the study guide, and then I'm gonna try and put together a practice test. Practice test.
Is the most important information that you want us to focus on the stuff you have highlighted and red? For the most
Yes.
part?
That's exactly right. I mean, there's a lot of information there. And I don't want to highlight everything, but I'm trying to select the key terms there to help you remember things. I mean, definitely look at the other words to kind of put things into context, to make things more understandable. But yes, try and focus on those highlighted words there. Good questions. Other questions about the test coming up, or about this week? All right, you're here to learn. All right, let's take a look at. Let's kind of remind ourselves what this chapter is about. So, can anyone kind of give us like a one sentence? What is this chapter about? No idea.
Microbial growth in the laboratory of nature.
Very good. So again, when we're talking about growth of these microorganisms, we're talking about what? Increases in cell number. That's right. So not so much about increase in size, but the numbers, right? And then a lot of what we know is based on growing microorganisms in the laboratory. So in the lab yesterday, what did we call those circular masses of bacteria that were growing in your petri dish? That was a colony. That might make a good fill in the blank, right? Like a colony. Okay. And then we're starting to learn about some of the conditions under which these microbes can grow in. So I'm trying to think of, let's just start off with this one here. So temperature, for example. We got a little bit of an introduction to that in our lab, where we isolate microbes from your own body. So just as a reminder, we're looking at this Celsius or centigrade scale here. What number corresponds to your normal body temperature? Over here? No, not over here. You remember? What was that incubator? You remember the incubator that had the 30, it's supposed to be 37. I know the incubator is a little bit off there, but 37 is your normal body temperature right there. And then just for reference, what temperature does water freeze at? 0 and below, yep. And what temperature at which water boils? 100, yep, at the normal atmospheric pressure, that's right. So that kind of gives us some reference points there. Okay, and you're gonna see we have these 5 different categories depending upon what range these microbes prefer to grow at. So what does it mean if we have a higher versus a lower? What does that mean?
The peak is its like most growth.
That's right. So like you think like the best growth rate for these hyperthermophiles is what temperature roughly? It's around 100. That's right. So that's how you would interpret the graph here. All right. So we have these 5 different categories. So starting at the top, we have our psychrophiles. They can actually grow at temperatures at or just above freezing. So we have highlighted the Arctic and Antarctic, for example. So would these microbes be able to grow in our climate, in our region of the world? Well, maybe today. It's pretty cold out. Okay, so when you were a kid, did you ever go outside and eat snow? Yeah, looks like ice cream. No? Okay, don't eat snow. Okay, because there are microbes that can actually grow in that stuff. And remember we talked about there's microbes in the air. So whenever you have precipitation, the rain, snow is bringing that stuff down. Okay, so snow, I know it looks really pretty, looks really clean, but it's loaded with microorganisms, so don't eat snow. Okay, then we have the psychrotrophs. Psychrotrophs over here. There's a little note here that they're responsible for spoiling foods that are placed in the refrigerator. So let's see. So you can kind of see, here's our psychrotrophs. Okay. Anyone know what degrees centigrade or Celsius are most refrigerators set at? Lower. Let's try again. Lower.
10?
Lower.
Are you talking Fahrenheit or Celsius?
We're talking about Celsius.
Okay.
2.
Close. It's actually 4. 4. So you kind of know what that temperature is when you open up the refrigerator door. So we're looking at around here for the refrigerator. Okay, so it's within the range for the psychrotrophs. So they don't grow very fast, right? Because we're down here on the growth curve, right there. But nevertheless, they can grow. So whenever you put foods in the refrigerator, there's kind of a general length of time when you're supposed to eat the food or discard it. So can you keep foods in there for 6 months and then you still eat it? No, you're supposed to discard food after how long? Let's say you got some leftover food, you put in the refrigerator, and then it's safe to eat it for a certain period of time afterwards.
4 days.
So it's around 4 days to a week. That's right. That's right. So after a week, throw that stuff away because there are these microbes, the psychotrophs, that can grow, not very quickly, but nevertheless they can still grow in there. Okay, so why do we put food in the refrigerator? Well, it's to slow down microbial growth for food preservation. We are mostly concerned with the mesophiles over here. Those are the ones that can grow at body temperature at 37. Okay, so, we have this highlighted word pathogen, but remember that there's a lot of beneficial microbes that are in your body that are not pathogens, but those are also mesophiles as well. But the concept behind refrigeration is to try and put those foods at a temperature at which the mesophiles don't like to grow at. So that's the whole concept there. Anyone know who actually had the concept, the original thought about refrigeration? Okay, it actually goes way back to Francis Bacon. Francis Bacon, you may have heard of this guy. He actually took a chicken, and it was in the winter time, stuffed it with snow in order to keep it cool, chill it down. And he actually preserved the bird. I think he actually got really ill afterwards. So that's kind of a funny story about Francis Bacon, coming up with the concept of chilling foods for food preservation. All right, we talked about the mesophiles, which include, your helpful or microbiota, as well as the harmful pathogens. But then let's look at the last two categories. We have the thermophiles and the hyperthermophiles. Okay, so thermal refers to temperature. Philes refers to they love it, they like it. So they like living in these hot temperatures. The thermophiles can actually live in very hot, near boiling waters. The hyperthermophiles can actually thrive in boiling water. Okay. All right, if you were a bacterium, what category would you be? You would be a mesophile, that's right, because you like 37 degrees. All right, any questions about this one? Okay, so it might be a good matching kind of question, thinking or labeling or something like that, so think about that. All right, moving on to from temperature to oxygen. So more categories kind of gives us an idea of what the preferences are for microbes with regards to oxygen. So what we're looking at here are pictures of like test tubes, and the opening is at the top, but they have like a cap on it. The cap is designed to actually allow oxygen to flow into the tube, even though the tube is covered. Air can still get in at the top. So the way that the tubes are set up is that the top of the medium is exposed to air. So there's high concentrations of oxygen at the top, and as you move down, there's less and less oxygen that will diffuse down. So the very bottom has very little oxygen. So it's kind of like in a swimming pool where you're at the surface of the water, lots of oxygen. The deeper you go, less and less oxygen. Same concept here. And then the idea is that, try to imagine for these five test tubes, bacteria was added to it, and then later we want to see where do the bacteria prefer to grow. Do they prefer to grow at the top of the tube, somewhere in between, the middle, or maybe do they prefer to grow towards the bottom? And that'll give us an idea of their oxygen preference. So starting on the far left, we see bacteria growing at the top. So that means that they prefer oxygen. They need oxygen. And they are referred to as the obligate aerobes. Okay. And then moving to the tube next to it, we add the facultative anaerobes. So anyone remember what we mean by anaerobes? Anaerobe.
Without oxygen.
They can actually grow without oxygen. And then we're going to see that term used in these two tubes here. They can grow without oxygen. That's the reason why they can grow towards the bottom of the tube. But then let's look at the word before. We have the term obligate and facultative. So obligate means that that's the only conditions at which they can grow. So they can only grow if there's very little or no oxygen, or they can only grow if there is oxygen. But facultative means that they can, but they don't have to. So look at this tube here. Where do you see most of those red colored bacteria? Are they mostly at the top or at the bottom of the tube? Look at this one here. You see more red bacteria towards the top or the bottom of the tube? Towards the top. So the bacteria, they prefer oxygen, but they can grow in the absence of oxygen. They can do both. Okay. All right. Then we have the microaerophiles. They like oxygen, but not as much oxygen as our obligate aerobes. And that's the reason why they're towards the top, but not at the very top of the tube. And then the last category is our aerotolerant anaerobes. So anaerobes means that they can grow in the absence of oxygen, which is what we see at the bottom, but we also see them growing equally well at the top. So they have no preference. All right. If you were a bacterium, which of these 5 categories would describe you? You would be what? You would be an obligate aerobe. That's right. You need oxygen. All right, any questions about this one? Okay. So this might be a good matching question too. Like match the name of the category, with how well they can grow. All right, so we learned about temperature categories, oxygen categories, and now we're looking at pH. So this time 3 different categories based on what pH range that they prefer. The last time we reminded ourselves pH scale goes from 1 to 14, 0 to 14-ish. So those that prefer to grow near a pH 7 are referred to as neutrophiles. So they like pH 7. Those that like to grow below 7 are the acidophiles. They like acidic environments. And those that like to grow above 7, that like basic conditions, are the alkalophiles. So remember, alkaline means basic. Okay. All right, which one are you? Which one do you, for the most part, I know your body's got some different stuff going on, but mostly your cells prefer what? Mostly neutral, that's right. Yeah. Okay, so that's not, I think that's pretty clear there. All right, we're trying to emphasize the importance of controlling microbial growth versus killing microorganisms. And we've learned that you can control microbes, from growing on things like food, using refrigeration. You can also control microbes by controlling water content. So they need water to grow, and if you remove that water, they can't grow. So, like on foods. Can you think of an example of a food where water has been removed from in order to preserve it?
Jerky.
An excellent example, beef jerky. Okay, why do they do that? Well, they did that to preserve it, that's right. And then, similar kind of concept, you can take meats and smoke them. You just slowly remove the oxygen, and that will preserve it. Okay, how else can you remove water? In addition to using heat. Well, you can remove water by adding salts and sugars. So pretend that this is like a bacterial cell, and these little green dots are either salt or sugar. And then you're going to see that there's a high water concentration inside the cell versus outside of the cell because you have so much salt or sugar here. And so there's more, because of the higher water concentration inside than out, more water will exit the cell than enter. There's a name for this process here. The diffusion of water is called osmosis. That's right. So by osmosis, water flows out of cells and basically kills the cell. So by heavily salting, like meats or whatever, as well as adding lots of sugar, you can prevent microbes from growing. Let's see if we can think of some foods that are heavily salted. Ham is a good example. Who said that? Okay, good. So we got ham. And then think of something, another kind of food where there's a lot of sugar added to preserve it. Jelly is the answer I was thinking of. Wow, you're doing really well today. Because on Monday, people ask, like, how do I get an A in the class? And you get an A by reading my mind. Because if you can read my mind, you'll know what the questions are, right? And you're doing a really good job today of reading my mind. All right, here's some other fun things here. we have a couple of categories for bacteria that actually like or can live in these salty conditions. So we have a halotolerant. They can live on our skin. You know, as we perspire, our skin does become salty. And we had the halophiles that can actually live, you know, near, you know, bodies of salt water. And they're actually adapted to living under those conditions. So that's kind of interesting too. Okay. All right, what's next? All right, I included a couple tables here from your book because this might be a good study. You know, don't write all this down now because there's too much information, but this might be a nice summary table to look at as you are preparing for Monday's test. So we have like different temperature categories, oxygen categories, pH, and terms related to water availability. So maybe, you know, come back to these. You may have them already in your notes, but maybe use this to supplement your notes. All right, we got a couple more categories related to, this time it's related to nutritional factors. So we're initially looking at what are these microbes using for their carbon source? Like, where are they getting their carbon? Remember that all life form on the planet that we know of is a carbon-based life form. So we use carbon as a building block to make different compounds. And that's true for microorganisms. So the question is like, where, you know, what is the source of the carbon? So we have two different terms up on the screen there. One is heterotroph. So the carbon that heterotrophs use are coming from organic compounds that have carbon. So there's only 4 classes of organic compounds. Okay, can you think of what those 4 are? Four different classes of organic compounds that have carbon. Who's in genetics? Some of you are in genetics, aren't you? Okay. What are you studying in genetics? This is that.
Carbohydrates.
Okay, so we got carbohydrates, that's 1 of the 4.
Phospholipids.
Lipids is number 2.
Amino acids.
Proteins, composed of amino acids, and nucleic acids, the topic of genetics, right? So heterotrophs are getting their carbon when they eat, when they consume those substances. You know, proteins, the lipids, the carbohydrates, nucleic acids. But then we have autotrophs, so they're getting their carbon not from those organic compounds, but from CO2 that's in the air. Okay, so you're thinking, what kind of organism here? Like, what would be an example of an autotroph? Think about a living organism. It'd be like a plant, that's right. And then if we're thinking about microorganisms, we're now thinking about algae. You're reading my mind. Very good. Algae and the cyanobacteria, that's right. All right, good. So we got carbon source, and then we have, this table here describes like what are they using these elements for, like to build, you know, what are they building? So we talked about some of these things already. They're building amino acids for proteins, lipids for membranes, you know, nucleic acids for DNA, RNA, you know, sugars for polysaccharides, or like cell wall material, for example. So that's the reason why they need these different elements. Some of these things are required for enzymes to function properly. Okay, let's just keep going here. Let's look at energy sources. So we talk about carbon source, but what about energy source? Okay, two categories here. The energy can come from sunlight. So we have phototrophs, or they could come from different chemical substances. So we have chemotrophs. We have phototrophs and chemotrophs. Okay, so you're thinking, well, I'm not a plant. And you're right, you're not. You are therefore a chemotroph. All right, next slide. It's going to combine these two categories together. It's going to combine the energy source with carbon source. So we're going to look at 4 different categories by combining carbon and energy sources together. So this is a fun one here. Let's see. Here we go. Okay, 4 categories based on carbon source and energy source. So remember, photo means light, so that means they're getting their energy from sunlight, solar energy. Chemo means that they're getting their energy from chemicals. So it could be inorganic substances, so we get the term litho, or it could be organic chemicals, so we have the, you know, we have the term organo for that. So remember that organic means that they're originating from living organisms. They're either carbohydrates, lipids, proteins, or nucleic acids. That's what we mean by organic. Inorganic are the non-living things. Okay, and then the second part of that is the carbon source. So if they're getting their carbon source from like CO2, they're an autotroph. If it's from an organic compound, they are the heterotrophs. But then we have the litho-autotrophs, meaning again the inorganic, the autotrophs meaning from the CO2. Okay, so we can kind of combine those different concepts together to make those 4 terms. Okay, so which one are you? Are you a photoautotroph? No. How about a photoheterotroph? Is that you? Nope. All right. How about a chemolithoautotroph? Is that you? No? Okay. I guess by default, you are a chemo-organo-heterotroph. Because you obtain your energy as well as your carbon from the organic compounds that you consume. Okay, so like you, there are a lot of bacteria that are chemo-organo-heterotrophs. All right, any questions about this one here? Okay, good. Now we're going to be learning about more about how to grow these microorganisms in the laboratory. We have two general categories of substances that are placed in either petri dishes or in flasks where you can grow these microorganisms. We have two examples, and we'll see some more on the next slide here. But we have what's called a complex media. It's made up of a bunch of different chemicals, but the exact composition can actually vary. It's not really known exactly what's in there. So like one example of that would be like the hay infusion. If you take hay from different farms, they could vary quite a bit. And so what you're going to end up with can also be highly variable. But on the other hand, we have our chemically defined media. This is like when you know exactly what and how much you're adding into to make these different formulations. All right, so let's take a look at examples of the two. So for the complex medium, again, the exact composition is not known. So in this example here, they're using beef extract. So depending upon the animal, what you get can vary. It's kind of like when you buy a steak. Sometimes it can be really good, sometimes it's not so good. you never know, because you're working with a natural product there. Okay, let's look at our defined media here. All of these are chemicals that you can purchase and you can weigh out, and so you know exactly what the composition is. Okay, so that's what they mean by chemically defined. Now, in the laboratory, in the petri dishes that you are using, you're using a formulation called tryptic soy agar. So you know that agar is that solidifying agent from algae. Tryptic soy refers to soy protein that has been digested with the trypsin enzyme. Okay, so which of these two, either complex or chemically defined, do you think is tryptic soy agar? What do you think?
What are the clues again? What's the logic again?
Okay. Tryptic soy agar, soy protein. Are you familiar with soy protein from the soybean? Okay. And then digested with the enzyme trypsin.
Would it be complex now?
It is complex, that's right. We don't know. I mean, the soy protein that you're getting can vary depending on the farm. That's right. All right, now we're getting into examples of different media that can be used to grow and cultivate microorganisms. So some are referred to as being selective. So selective means being choosy. Okay. And remember that when you were looking at your petri dishes on Tuesday, some of you were able to identify different types based on size, shape, and color. Okay, so there's a lot of different kind of microbes that you can grow in a petri dish. But let's say you're only interested in one kind. You can use depending on what the bacterium is, you can use what's referred to as selective media that will actually promote the growth of one kind, but not other bacteria. The example here is with the McConkey agar, and this one is designed to promote the growth of coliform bacteria. Coliform refers to a part of your body. Your colon, okay, your intestinal tract as well. Microbes that grow in there, which includes E. coli, Escherichia coli. So the way that this one is set up is that you can have a whole mixture of bacteria, but only the red ones will be favored. They'll be selected to grow on that dish. Other bacteria that are not coliform bacteria, will not grow as well or won't grow at all. So that's what they mean by selective. So if you're interested in testing water, does it have E. coli contamination? You can use this kind of a medium here to look for just the coliform bacteria, and you're not really concerned about other ones. Okay, so we have selective media, and then we have what's referred to as differential. So differential means you can distinguish one from another, right? And in this example, we actually have an agar that's colored red because it contains blood. Heme from hemoglobin is in the agar, and that gives us the red color. And there's actually some bacteria that have enzymes that will break down the heme. So they'll take the red and break it down, and now you don't see any red anymore around the bacteria. And the ability to break down that hemoglobin, that heme, is called hemolysis. So here's a medium that is not selective. So you can actually have different kinds of bacteria growing, but it does allow you to distinguish the ones that can break down heme from those that do not. So that's what they mean by differential. You can differentiate one from another one. All right, are you up for one last slide? Yeah, kind of, sort of. All right, let's see what we got. Maybe I'll skip this one here. Okay, let me actually go back. See if I can go back, and we're gonna end with this slide right here. All right, what does obligate anaerobe mean?
Requires oxygen.
Anaerobe.
Can't grow with oxygen.
Can't grow with oxygen. Okay. But wait, this room is filled with oxygen. What do they do? So if you have an obligate anaerobe exposed to the room air, what do you think will happen? It won't grow. It'll die. Okay, so it's really hard to study these kind of microorganisms when our rooms are filled with oxygen. Fun thing, my nephew actually is a scientist working in Japan, and he works with these obligate anaerobes. He took these core samples from the depths of these Canadian lakes. And they're packed in mud, so that means there's not a lot of oxygen in there. And he's able to grow these things in the laboratory and discover new organisms that we didn't know about before. And one really interesting thing about these obligate anaerobes is that they are actually associated with areas where crude oil is located. Okay, so there's like these microbes actually like to grow where you can actually drill and find oil. So knowing that, some of these oil companies actually hire microbiologists to help them find sites where there's high probability of drilling and discovering new oil reserves. Who would have known, right? All right, so we're going to end on that note about some of the beneficial aspects of knowing about or having knowledge of these microorganisms. All right, we're going to finish up this chapter on Friday. I'm going to try and get a practice test out to you, and then we'll get ready for Monday's test. So have a great day, everyone.